Hello. Today we're diving into one of life's greatest secrets, DNA replication. This is the process that's happening inside our bodies every single second. And it's the reason we grow, our wounds heal, and, well, why we stay alive. So let's get started on this incredible journey. So it all kicks off with this one simple question. How does one cell become two? You know, when your skin heals from a cut or when a baby grows, it all comes down to cell division. And at the very heart of that process is making a perfect copy of our genetic blueprint. All right, so first things first, let's get a handle on the basic principle, how this code of life actually gets copied. So what exactly is DNA replication? Well, it's the process where a cell makes an identical copy of its DNA. This is absolutely crucial because it makes sure that when a cell divides, every new cell that's created gets the complete set of genetic instructions. You can't have any missing pages in the instruction manual, right? Now, the core principle of this whole thing is called semi-conservative. I know, it sounds a little complicated, but the idea is actually super simple. Imagine you have a necklace made of two intertwined strands. To make a copy, you pull the two strands apart, and then you build a brand new strand alongside each of the old ones. And the result of that is that every new DNA molecule is made of one original or parent strand and one brand new strand. As you can see right here, that's exactly what semi-conservative replication means. Half old, half new. So why is this process such a big deal? Think about it. Everything from us growing bigger to our skin healing after an injury and even passing our genetic information to the next generation, it all depends on DNA replication. It's what keeps our genetic code stable and secure. Okay. Now that we've got the basics down, let's look at the three main stages of this process. It works kind of like a molecular assembly line. So, these three stages are initiation, where the DNA unwinds, then elongation, where the new strands are actually built, and finally termination, where everything is wrapped up and the process ends. We're going to look at each one of these. During the elongation phase, something really interesting happens. Because the two strands of DNA run in opposite directions, the cell has to use a clever strategy to copy both at the same time. One strand, called the leading strand, is built continuously, all in one smooth go. But the other one, the lagging strand, is built in little chunks called Okazaki fragments, which are then stitched together later. But you know, none of this happens by itself. This entire job is carried out by the real heroes of our story, the enzymes. So let's meet our molecular team. First up, we have helicase, which acts like a zipper, unzipping the DNA. Then there's primase, which creates a starting point to get the process going. Next up is the star player, DNA polymerase. This is the master builder that adds the new nucleotides and even checks its own work. And finally, we have ligase, the gluer that joins all those Okazaki fragments together on the lagging strand. You know, probably the most amazing part of this whole process is its incredible accuracy. Let's take a look at how it manages to avoid making mistakes. Just look at this number. On average, there is only one mistake for every 1 billion nucleotides copied. That is unbelievably precise. It's like copying a book with 1 billion letters and only making a single typo. So where does this incredible accuracy come from? Well, two main reasons. First is proofreading. Our master builder, DNA polymerase, is constantly checking its work as it goes. And if a mistake still slips through, there's a second team of repair enzymes that comes in to fix any leftover errors. Whew, that was a lot of information. All right, let's quickly go over the most important things to remember, our key takeaways. To remember the main enzymes, here's an easy trick for you. HPPL. That's helicase, primase, polymerase, and ligase. These are the 4 key workers that make this entire process possible. HPPL. So what are the main points? Remember, replication is semi-conservative, making one old and one new strand. The process uses a leading and a lagging strand, and it's an incredibly accurate process that's all controlled by enzymes. And now I want to leave you with this question. We've seen just how accurate this process is, but what happens when it does make a mistake? What happens when an error slips past the proofreading and repair systems? These rare errors can lead to mutations, genetic diseases, and even the process of evolution itself.